division, fission, mitosis, and meiosis. And today's class, this is year 3 of science, all right? And just to get started, we are going to do a couple of things, okay? First of all, I want to call your attention here in the lesson on the Family School Online. I listed here some of the videos that we have for the lesson. And here there's important instructions about how to build homemade Petri dishes, which you will be using. Okay, these are the instructions that you could use. This would be one of the possible assignments. Okay, so this assignment would take about a week to complete. But the idea is for you to make different cultures of bacteria and grow these cultures. So it has instructions on basically how to prepare a Petri dish with food for the bacteria. Then with a swab, you get the bacteria in and you feed it there to the food.
What's a Petri dish?
So a Petri dish.
What's a Petri dish?
Excellent question. Let me go and show you one. All right. So this is a picture of a Petri dish. So it's basically a plastic container with a lid. Okay.
Is it edible?
Use any. No. This is a good question. Okay. I'm really glad that you asked.
I tried some already.
So you try what to eat? Some of this? So
yeah, it tastes good.
It tastes good? No, so the idea here is that you put a plastic container with a lid and you prepare inside the Petri dish a sugary medium, basically a sugary place for the bacteria to eat. So it's gelatin, water, sugar, okay, you have bouillon cube or something like that, and then in a small container, all right? And then you put a bacteria on it, and then the bacteria start eating and multiplying and growing, okay? So this is not something that you would want to really eat.
I know.
Okay.
Who would have an idea to eat that stuff?
Well, so there are some bacteria that are good to eat and that we eat kind of normally. Like for example,
what bacteria is good to eat?
The bacteria in blue cheese. Okay, so for example, blue cheese. Okay, the blue stuff in the cheese is bacteria. Okay, yogurt is also made out of bacteria. Okay, so there's a lot of bacteria that we actually eat, and it's good, you know, but we don't know which one is which. Okay, just so I'm not going to ask you to eat this bacteria that you grow. The idea is that you will have, you know, your Petri dishes here, and you grow the culture of the bacteria to watch it. Okay, you can probably put it in a microscope and see if they are different or the same. All right. Mm-hmm. Does that make sense, Josh? Thank you for asking. Good question. Okay.
Yes. Yeah,
so that, All right. So that is what we are going to do for one of the assignments, one of the choices of the assignments. There's a few others.
Okay. Okay. Okay.
Yeah.
Okay.
Bread also, you know, has the bacteria, okay, that we eat, has both yeast, that this is a fungi, and bacteria in it. And when the bread starts going green, okay, that is actually the bacteria taking over the bread, okay? Kind of starts reproducing a lot and eating the bread. So if you put a piece of bread, especially if it's like a wet piece of bread in a bag, you can start seeing the bacteria growing. Anna asked, is bacteria the same as mold? No, my understanding, and let's go on. You know, these are good questions, okay? But usually, what I like to do when I have this type of questions is basically just search for the answer. Okay, so I'm going to go here to Wikipedia. My understanding is that the mold is also a fungus. Okay, so let's say mold. All right. So mold is a fungus that grows in the form of multicellular filaments called hyphae. Okay. In the next lesson, we are going to talk about fungi. All right. So what is the difference between then a bacteria and a fungus? What do you remember about that? You know, remember that we had very big, broad classifications of cells. We call some cells that were one type and another cells being the other type, okay? You remember that this is probably in the first, second lesson, all right? Not this. Okay, Emily says fungi is eukaryotic. Anna says prokaryotic and eukaryotic, okay, cells. That is correct. And so bacteria are prokaryotic. And molds that are fungus are eukaryotic, all right? So fungus have nucleus in the cell, and the prokaryotic cells don't have nucleus, all right? Okay, so very important questions, but now you see how everything starts kind of getting together, all right? And now you can understand when you ask the difference and the question between one and the other, what's the difference, okay? So that is a very, very important thing, all right? And here it says that molds sometimes, okay, are the ones that cause food spoilage, all right? And so sometimes, you know, like in the bread, it could be that it's a mold growing and not a bacteria. If you were to have a microscope and you look at that, how could you tell if what you are seeing is a mold or a bacteria? What would be the thing that you could look for to distinguish between a mold or a bacteria? What do you think you can look for? Anna says the mold would have a nucleus, right? Yes, that's right. That is absolutely right. Jacob says spores or spores. Yeah, so the mold, the hyphae here, okay, is kind of like that fuzzy, hairy type of organization of the mold, with characteristics of the mold, all right? But what you would look for in the microscope is for the nucleus. So if the cells have a nucleus, then it's a mold or some sort of eukaryotic cell. If they don't have a nucleus, then you know that it's a bacteria, okay? Prokaryotic cell. Very good, excellent. All right. So let me do a little bit of review here before we get to the first concept. So do you remember that when we were talking about the kind of the reproduction or the synthesis of proteins, we talked first about the process of transcription and how DNA was transcribed into RNA. And then we talk about how the RNA was translated into amino acids. Well, here in this video, we are actually seeing the mechanism of how DNA is replicated. Before the cell can divide, all the DNA in the cell has to be completely replicated. It has to be copied all the way. And the way to do that is by using these proteins and doing this process that we see here in this animation. And it seems kind of complex, so let me explain a little bit about what is it that we are seeing here. First of all, this blue protein here is the protein, I think that is called something like helicase or something like that. I don't remember exactly, but the idea of this protein is that it inserts itself into the DNA molecule and it splits the weak hydrogen bonds between the nuclear bases, okay, the nitrogen bases, between the A and T and the C and G. And so it separates the two strands of DNA. One of the strands then is then separate, and here we have the same protein that we use to do the transcription, you know, DNA polymerase. So this protein basically starts getting nucleotides and attaching them and recreating the double helix that we see here. The problem is that the molecule of DNA is not symmetrical. It's not exactly the same one way or the other. And so one strand can be directly replicated. The other one needs to be flipped before it can be replicated. And so this is what the molecule is actually doing. See how a part of the, okay, let me play that part again. See how a piece of the DNA is kind of flipped without being cut, okay? It is flipped upside down in order to make it the same way that this other side. And then the DNA polymerase can start adding the nucleotides, you know, completing the double helix, all right? You see how it goes? So this is an amazing process of flipping the DNA in order to be able to complete it. See how intricate this machinery is in order to do that. Now, this happens extremely fast, about 2,000 nuclear bases per second, okay, can be replicated this way, okay. So that is the process of DNA replication. Yes, it's like a jet engine, really. So it is really an amazing and very, very fast process of how the whole DNA is replicated. So this is different than transcription. In transcription, we just transcribe one strand of the DNA into RNA, all right, and it was for the purpose of creating RNA, and then the other one was the process of translation, which was translating the RNA into amino acid chain for the protein synthesis. In this case, we are replicating the full DNA in the molecule, okay? So now we are going to go to academic concept 1, and Patrick, would you like to open your mic, okay?
Hi.
Take us there. Go ahead.
Okay, so there are pretty much, we reproduce our cells by splitting them, but there are 3 different main types of cell reproduction. We have, let's see here, see if I can remember this. We have mitosis, cytokinesis, I hope I'm pronouncing that right, and I think meiosis.
So what were the three again?
Mitosis, cytokinesis, and meiosis.
So meiosis and cytokinesis. Cytokinesis is one part of the meiosis process. Okay. So it's one of the phases of meiosis. All right. So the three are binary fission. Okay. Binary fission, and then meiosis. Okay, let's say mitosis, and then the other one is meiosis. Okay. What did you learn about these different processes, Patrick?
Well, I learned that one of them is for when we are creating new humans.
Mm-hmm.
One of them is for when we are growing. And one of them is for bacteria when they are trying to reproduce.
Excellent. Good job. Yes. And which one is the process of mitosis, okay, is for what? Which one does mitosis do?
I believe mitosis is when they split.
Mm-hmm.
And meiosis, I'm very dyslexic. Meiosis is when we are when we are creating new life during procreation. And the last one, which I do not remember the name for, is for when the bacteria is split.
When the bacteria split, that's right. That is binary fission. Okay, fission. So the word fission sometimes is confused with fusion. Okay, fusion and fission. So we need to make sure we don't mix those two words. Fission is to separate something, to divide something. All right, to really split it. Fusion is to put it actually together, okay? And so in this case, the prokaryotic cells use a binary fission. Mm-hmm. All right, excellent. Now, in the academic concept here, it says that the process of mitosis, okay, basically has, three phases, all right? Interphase, mitosis, and then cytokinesis. Those are the three stages in the cell cycle, okay? So the cell cycle, we have those there. Could you describe what is interphase? What did you know about interphase?
Interphase is when the cell is expanding, preparing to split, but not even close yet. It's just gaining mass so that it can split. And then mitosis is when it is going through the process of just getting actually ready to split. And then cytokinesis is when they actually do the splitting and split.
When it actually divides.
Mm-hmm.
Yeah, excellent. In this graph here, it shows the cycle, okay, the cycle, and this kind of shows also the amount of time that the cell spends on each part of the cycle, about kind of proportional, all right? So which parts of this cycle would be then the interphase?
The very middle.
Hmm?
The very middle. The purple thing at the arrow. It's like an outward spiral.
This whole part, okay, so this whole part of the cycle is interphase. So interphase comprehends all these parts of the cycle. So if, let's say that this is about, 24 hours, all right, about 20, 22 hours of the cell would be during interphase. Okay?
Okay.
All right? An interphase has 3 phases. What are those?
G1 phase, oh gosh, it's popping up. G1 phase, S phase, and G2 phase, which is, if I can get the stupid thing to stop popping up. Dang it. Which is cell growth, cell replication, and preparation for mitosis.
That's right, yeah.
Sorry, the cell cycle download button keeps popping up.
So where does when does the cell actually does its work? So let's suppose that this is a cell in the stomach or a cell in the blood vessels. Okay. When does the cell actually produce does the transcription of DNA and does the protein synthesis and so on?
Sorry, my brother was bugging me.
Okay.
What?
So during what phases of the cycle does the cell perform its work? Like doing its job.
I believe that it actually does its job during the cell growth and like in the beginning part of the replication and
so
the S phase.
It would be here during the G phase, G1, and the G2 phase. Okay.
Yeah.
What happens during the S phase?
It replicates.
It replicates what?
The chromosomes.
The chromosomes. So the things that we saw in this video of how the DNA was replicated, that happens here during the S phase. Okay? And it has to have the whole DNA replicated in order to be ready before the cell can divide. Okay? Excellent. Okay. And then we have different other phases for the rest of mitosis. Okay? We have this prophase, metaphase, anaphase, telophase. All right? And then at the end, it does the cytokinesis. So what do you know about each one of those phases of mitosis?
Well, I know in the prophase that the DNA is sort of condensed into the chromosomes, and each of the centrosomes are moved to the opposite side of the cell of the chromosome. During the metaphase, they actually start to move away from each other and latch onto the chromosomes. And during the anaphase, they pull the chromosomes apart. And the telophase and cytokinesis phase is when the cell splits and becomes a different cell.
Yeah, that's right. What would happen after that?
They would reproduce the other half of the chromosomes.
So it would go back into the G1 phase, okay? Both of the cells would start growing again, okay? And then they would do the S phase, the G2 phase, and then divide again, all right? Okay? So that is kind of how it would repeat. That is why this is like it's called a cycle, because it happens kind of all the time. Okay. Anna is asking, do cells ever stop dividing? What do you think, Patrick?
Well,
Do cells
I
ever
think
stop
that
dividing?
cells even divide after people die because their hair and fingernails grow. Well, the the dead cells move up. So I think that they do stop dividing after you die. That's my opinion.
Okay. So it is interesting to know that so most activity actually when the organism dies, most cellular activity just stops. All right. So when the cells split, they can continue splitting. All right. Now there are some cells that exit this cycle and they stop dividing. All right. So for example, during the time when a baby is developed inside the mother, okay, there's a lot of activity and of course there's a lot of cell division, and this cycle is happening all the time, and the cells start the process of differentiation. So at the beginning, you know, you have the zygote, that is the mother cell, and then that one starts dividing through mitosis. But even after just a couple of days, so even a couple of days afterwards, the cells start dividing, but they start being different from the other cells. And so there are some cells that become bone cells, and others that become eye cells, skin, and neuron cells, and so on. Okay, so that is really how it happens. There are some cells that exit this cycle and stop splitting. For example, the neuron cells, the cells that make our nervous system, after they get developed, they don't split anymore.
And that's why being quadriplegic is so bad, because they don't heal.
That is exactly right, Patrick. Yes, because the cell, once it's damaged beyond repair, it cannot reproduce another cell of the same type. All right. So there are some cells that stop the cycle of mitosis. Okay. So that is very, very interesting. That is also, for example, why you know, I've heard that if a horse, okay, breaks a leg, the bone won't repair itself, okay, because the cells won't go into mitosis. All right, and so that is one of the things that can happen. There are certain cells in different organisms that stop replicating. Okay. Also, with age, the process of mitosis starts slowing down. Okay, and that is one of the reasons why we age. If you think about it, you know, as a child is growing, you know, the process of mitosis is happening a lot, very, very fast, very frequent. But as the person ages, the metabolism slows down, and part of that slows down metabolism is that this cycle also slows down. All right. There is one case, Patrick, when the cycle kind of keeps going and starts going out of control, and the cells continue doing mitosis uncontrolled. Okay.
Isn't that cancer?
That is cancer, exactly. Good job, Patrick. That's right. That is what happens with cancer. So cancer is basically when this process of mitosis continues without stopping, you know, and that is why it's a dangerous thing. Mm-hmm. Ryan, you have a question? Go ahead and ask your question. And you are unmuted. Go ahead.
Okay. So I was reading through the lesson, and it said that cells take about 24 hours to make a new cell. Does that work with, like, if you had a bug, would the cells be, would it take longer or something?
If you have what? Sorry, I couldn't hear that correctly. What is the question again?
If you had like a bug, would the cells in the bug, would they take longer to make new cells?
Oh, the cells, the bone cells? Mm-hmm.
No, and the, like if you had a bug, or a fly or something, would the cells take longer for them to make new cells?
Okay, I understand everything except the one word in your question. If you have what?
A bug or a fly or a caterpillar or something like that.
I still don't understand the word. Could you write it down in the question? In a bug, you mean? A bug?
Yeah.
Okay. Okay, so what do you mean by a bug? Let's be more precise, because I think that that term is used, you know, too generically.
Like, if with the cells, well, it said in the lesson that it takes about 24 hours for a cell to make a new one. For if you had a bug or something, would that take longer? Make a new cell
so if what you mean by a bug is a bacterial or a viral infection, yes, it could take longer. Okay. So what happens is that if you have a bacterial infection, all right, then the organism it sends the signals to the cell to start focusing more of the energy into fighting that bacteria, okay?
Okay.
And so it starts producing white blood cells and other things to fight specifically that bug, okay? So we need to be a little bit more precise in what we mean, all right? So if it's a virus, the interesting thing, we are going to talk about viruses in another class, but the virus actually accelerates accelerates the cell division for the cells that it infects, and then the body has to create the antibodies to recognize, okay, that is not a good, you know, a good cell, and it starts to fight against the viral infection. That is one of the reasons why viral infections take longer for the body to break down. Okay. All right. Does that make sense?
I have a question about.
Yeah.
Sorry. So I don't have a petri dish at home. Is there a way I could really experiment without it at all?
You can create a petri dish almost about anything. Okay. The only thing is that you need to make that sugar agar, you know, that sugar medium to feed the bacteria. And you can do it inside a plastic bag. You can do it inside a Ziploc bag. You can do it inside, you know, one of those little cups that you would use to put, you know, like condiments or something like that. All right. any jar, anything. All right. You don't need to have any professional stuff here. All right.
Okay.
Okay. Excellent. Good question. Good question. Okay. So let me see if we have, I think, a couple of videos here that show the process of mitosis. All right. So here we are seeing, how the cell divides. Let me play that part again, which was very, very interesting. Let's see that. Okay, so here we see how the chromosomes are already replicated. So this cell is already in which part of the process would it be? Okay, so if we go here to our table, okay, in what part of the process it would be? Let's go back to that, to the video here. Okay, so what is going on here? Do we see now the cell nucleus already? Patrick, are you still there with us?
Yes.
Okay, so do we see the nucleus of the cell? So apparently, the nucleus disappears. During mitosis, that is one of the things that happens, is that the nucleus disperses, okay? The nuclear membrane, okay, disappears.
I was going to say,
right.
is it the whole thing like this? Because,
Yeah, so this is prophase, okay? So this part, okay, here we see the chromosomes are compacted. You can see them that they are organized and condensed. They are kind of moving around, all right? And so that is the prophase part. Let's go and play it a little more. Okay, now, all right, here, this happened really fast. Okay, so let's go back and see prophase. Then the chromosomes align towards the middle of the cell. Let me see if I can stop it in time. Okay, when the chromosomes align, that would be the metaphase. And then when the chromosomes start pulling away, that's anaphase. Okay, and so that is the anaphase there. You see how they are being pulled apart, and half of the chromosomes go to one side of the cell, and the other one go to the other half. And then the chromosomes uncompress, they diffuse themselves, and then the two cells are back created, okay? And that is cytokinesis. Here we see the nuclear membrane being formed again, the cell pinching, and that is the cytokinesis part, when the cells actually divide. Here we see the kind of the animation of how the chromosomes are separated in anaphase. Aubrey says that it looks like a spider here. Yeah. All right. So, Patrick, anything else that you would like to add about what you know of mitosis?
Not really, no.
Okay. All right. Okay.
Right.
Good deal. All right. So thank you very much. You did very, very good. Anna, would you like to take us to the second academic concept here for binary fission?
Okay. So I guess this academic concept is about prokaryotic cells and how they divide by using binary fission. And binary fission, it says, is used by prokaryotic cells like bacteria, and is also used by a couple of eukaryotic organisms. these, let's see, hold on a minute.
Yep.
Okay, so it looks like in a prokaryotic cell, it divides the DNA, it duplicates itself, and the DNA attaches to different parts of the cell membrane, and then the cell starts to pull itself apart and kind of stretch out, and then it replicates its chromosomes, and then the cell will start stretching out some more, and the ribosomes and the plasmids, they replicate, and they go to the different sides of the cell, and then the cell kind of pinches and forms a cell wall, then it breaks off into two different cells that are identical.
That's right. Yes. So, do these prokaryotic cells have chromosomes?
Yes.
So actually, they don't. The DNA, that is one of the differences. No problem, okay, but that is one of the differences in prokaryotic cells. The DNA is just one circular strand of DNA. Okay.
Okay.
So all the genes for the prokaryotic cell are contained into just one circular, ball of DNA. Okay.
So is it still like a double helix? Oh, go ahead.
It is a double helix. Yeah, it's still DNA. It's the same thing, and still when it does the replication of the DNA, in order, before it can divide, it has to unravel the DNA, do a complete copy, and the copy is exactly the same as what we saw here in this video, So it still does the same, with the same proteins and the same process to replicate the DNA completely, okay? The difference is that it's not organized into chromosomes. It's just a copy. Sidney says it's just a copy. That's right. It's a copy of the full DNA, all right? In order to create a cell by fission, is an identical cell. And then once the cell is divided, then the DNA basically goes back into its kind of ball and starts, producing proteins and doing all the things that the DNA does. Okay.
Okay.
How fast is this process?
I think it took like 20 minutes, I think.
Yeah, under the right conditions, bacteria can replicate really, really fast. Okay. So it is really interesting that bacteria can replicate very, very fast in that. Let me see if we have, okay, let's see. So this is another video of mitosis, okay. But there was another one I'm going to see, I think that it was somewhere in here, that I had a link to a little video of fission. Let me see if I find it in Wikipedia. Okay. Okay. Where did I see it? There was a, let's do a Google for that. Really fast, okay. Bacteria dividing, let's try try that. Okay, let's see if that is a good one. So yeah, okay, so here we see how the bacteria divides. This is accelerated, but that is basically how it goes, and see how fast it grows. All right. That is what you would see in the experiment that you do when growing your culture. You are probably going to see kind of the division there of the bacteria really fast.
Wow.
In literally a few minutes, you can grow millions of bacteria. Okay.
That's
because
really
each
cool.
one is divided by 2, yeah. Okay. So that is interesting. All right. Yeah. Okay, let's see. So that is how binary fission happens. Okay. And Sydney asked, I am doing that? Aubrey says it is freaky, looks like something from my nightmares. So Sydney is asking, I am doing that? Okay. What do you think, Anna? Are we doing fission?
No, because we're eukaryotic.
That's right. We are eukaryotic organisms, so we don't do fission. We do mitosis. Okay, so most of the time, our cells are in this cycle of doing mitosis. All right, but there are some organisms inside of us, okay, that are actually doing that. What would those be?
I think like the mitochondria.
That's right. Mitochondria inside the cell divides by fission. Excellent. Good job. Okay. Also, inside of our organisms, we have a lot of bacteria. There's over 1 trillion bacteria in just our digestive tract, in the stomach and the intestines and everything, and they help us to digest food. So it's actually a very important type of bacteria. We provide a really nice environment for the bacteria, and they break down some of the food that we eat, okay? They eat some and we eat some, okay? So Sydney says, oh, I meant that I'm doing that in the experiment. Yes, you are doing that in the experiment, okay? The video was, I just pulled one video on binary fission. Okay. Audrey says, mitochondria is probably one of my favorite science words, just because it is fun to say it. That's right. Excellent. Okay, so here we see that now we learn about the 2 main processes of cell division. The mitosis, okay, most of the cells, all the eukaryotic cells go through mitosis, and then fission, which is mostly for the prokaryotic cells. Okay, so excellent. The third academic principle, thank you, Anna, very, very much. The third academic principle says that eukaryotic organisms reproduce children of the same kind, children organisms, through a more complex method of sexual reproduction called meiosis. Okay, so in meiosis, or meiosis, I don't know how it is pronounced. Okay, I'm a foreigner, so I'm going to say meiosis. All right, so it's meiosis. I don't know exactly. You tell me which one is correct. But in this process, the cell is basically, and the chromosomes are actually broken down twice. Okay, so and before the process is a lot more complex. It's a little bit more complex. But the interesting thing is that as the chromosomes align, okay, so here we have one cell with chromosomes from the parent, okay, so the blue ones, let's say, are the chromosomes from the father of this cell, and the red ones are the chromosomes from the mother of the cell, okay? So all our cells have 2 copies of each chromosome. That is why we have 46. We have 23 from dad and 23 from mom. And all our cells have the same copy through mitosis, okay? Through mitosis, they have the same copy of all 46, okay? So basically, normally, during interphase, we have 46 chromosomes. When we duplicate those during the S phase, okay, we double the number, and then during the separation, that is where we divide, and then each child cell will have 46 chromosomes again, okay? 23 from the mother and 23 from the parent. In meiosis, what happens is that the chromosomes first align, and then they twist until they start crossing over. And some of the genes of the father go to one side, and some of the genes of the mother go to the other, and so on. And that is what creates the diversity of eukaryotic organisms, okay? Then the cell is divided, and then the cell is divided again, and what you end up is, instead of dividing into 2 cells with meiosis, you end up with 4 cells that each one has half of the genetic information that the organism needs, okay? So meiosis is the way of producing the gametes for sexual reproduction. Okay, so men or males would produce a sperm that have just 23 chromosomes, and females would produce eggs that have also only 23 chromosomes. For the new organism to complete, you need the process of fertilization together with the process of meiosis. So during fertilization, you join a male gamete, a male sperm, with a female egg, and now you get the 46 chromosomes again, and you have a zygote or a fertilized egg, and that then would start the process of mitosis again in order to divide and create a new organism. Okay, so meiosis is a very, very interesting mechanism that the Heavenly Father created in order to have parents, okay, and to have sexual reproduction and the amazing variety in eukaryotic organisms, okay? Because if we were only reproducing by fission or by mitosis, we would all be the same. We would all have the same genetic information. But the process of meiosis is what makes the crossover of the genes and the transfer of genes from one generation to another and make everything different, and that is what makes us different, okay? So during meiosis, the genetic information of parents and grandparents is recombined to create an amazing diversity of the children. Does that make sense? Okay. Nathan, would you like to help us understand how this relates then to the gospel principle of tithing?
Sure. So the Rosberg principle is when we divide our increase with God through paying and tithing, He multiplies our blessings. So I will let the scripture just a minute.
Mm-hmm.
It's in the New Testament. It's about the widow's mite.
Yeah.
And so the story is that the Lord, Christ and his disciples are at the temple, and all the people are coming paying tithing. And then it says, and the chief seats in the synagogues and the uppermost, and devours the widow's houses. And it's saying that all the rich people are just. And there came a certain poor widow, and she threw in 2 mites, which make a farthing. And he called unto him his disciples, and said unto them, Verily I say unto you, that this poor widow hath cast in more in than all they who cast in their treasury. For they cast in their abundance, but she of her want did cast in all that she had, even all her living. So it's saying that it's not bad to put in a lot of money, but the priests and the scribes and Pharisees would do it to brag about it. Oh, I can put this much in, but that's not the point. But it's okay if you give a lot of money for the right reason, but if you are just doing it so you look good in front of all your peers, and that's not okay. And the Lord especially loves it when you give all you can. If you can only give like $5 a week for tithing, then that's okay with Him as long as you're giving all that you can.
That's right. Yes. And how does the Lord recognize our sacrifice?
Well, He always knows our wants and our needs and how we can afford. So some people might think that they can trick the Lord and say, "Oh, I can only give this much money because I need to do all this stuff for the rest of my money." But He's smarter than us, so He knows whether or not we're doing giving it all we can. But it's always important that we do.
Mm-hmm. That's right. Liberty is asking an interesting question. She said, so how do you keep the law of tithing if you don't ever make any money? Okay. So let's suppose you have someone that doesn't have any money. Okay. What can they do to keep the law of tithing?
Well, the law of tithing is 1/10 of your increase. So if you are not making money but you're getting more or something else, like let's say you make blankets and you make about 10 blankets, then you can help give that to the church and have that be to the poor, because in the scripture it says 1/10 of your increase. So it doesn't have to be money. The church appreciates that if you sell your stuff for money, but it's just 1/10 of your increase.
Yes, that's right, of your increase. Absolutely. Okay. So Liberty jokingly says if you have 10 children, you should give one to the church. So it is interesting because I always thought that, for example, for me, when I was going on my mission, giving 2 years, okay, or dedicating just 2 years as a missionary was kind of like tithing the first 20 years of my life. Okay.
I have a question.
Mm-hmm. Yes.
How do you pay? Do you pay tithe on your mission?
That is a very good question. Okay.
You don't really make any money.
Mm-hmm. So you are not making any money, and the funds that you receive from the missionary fund are already dedicated for the missionary work. Okay.
Yeah.
Plus even if it's your own money, you already have paid tithing on that money. Now you are just using the money for missionary work, okay? So normally you do not pay tithing during your mission, okay, unless you receive anything extra, all right? But one thing that I always thought of is to always pay a very generous fast offering, okay? So during my mission, personally, I didn't pay tithing because the funds were already dedicated for the missionary purpose. They had already been tithed, but I always paid fast offering, okay? Because frankly, you know, in many of the areas where I serve, I was probably more, you know, I had probably more money than many of the people around me, okay? And so I felt that, you know, I certainly could do that sacrifice and fast and then pay a generous fast offering. Okay. Aubrey says there is a proper name for that I cannot remember. Are you thinking about consecration? So the money is already consecrated, okay, or made sacred for a specific purpose. In this case, it would be for missionary work. Okay. All right. So it's not exempt, okay, so no one is ever exempt from paying tithing, okay, but we already, you know, we could already consecrate our time or funds for some special purpose. Or it could be that the money has already been tithed, okay. This is an interesting, have you had a chance, Nathan, of seeing this quote from Elder Oaks from when he was a child about tithing, his experience with his mother?
No, I haven't.
Okay. So
I know, I think I've heard it before, isn't it? How his father dies and him and his family are very poor and they can't afford a lot, but his mother always paid tithing, so they always had enough of what they needed.
That's right. That is exactly right. You remember correctly. Yeah. And even though she was a widow, okay, she says, you know, we have to pay tithing because we need the blessing. We certainly need the blessing. Okay. All right. Excellent. Well, thank you very, very much. One of the things that I want to kind of emphasize about this is that the same of how, you know, we were seeing this here, this bacteria dividing, you know, really fast and multiplying, the same way is how the Lord multiplies our blessings, really. When we obey His laws and everything, He makes the life increase. Okay, so that is really important to me. Okay, so Emmeline says, quotes President Monson saying, all of us can afford to pay tithing. In reality, none of us can afford not to pay tithing. That is very, very true. Very true. So Sydney says, in the Philippines, the Filipino missionaries used to spend as little money from the missionary fund as they could, and then they would send the rest to their families to help them. They got in big trouble for that. Okay, my dad told me this. That's right. So the funds that you receive as a missionary are consecrated for missionary work. And so if you have any extra money from your assignment, you can return it to the mission home, but it is missionary money. It cannot be used for anything else. So thank you for sharing that, Sydney. Good clarification. Anna says, I think that tithing helps to test our obedience to God's commandments. Our obedience, our faith, okay? So, and it's a very fair, law of tithing, right? So, when we go on missions, I would not say that we are exempt from paying tithing. I would say that the funds that we receive are consecrated for missionary work. And so we always need to be careful that we use those funds because they are sacred funds, that we use them for that purpose. Okay. So, Andrew, would you like to finish the class with a prayer for us?
Yeah.
Would that be okay? Excellent. Yeah, we can.
All right. Dear Heavenly Father, thank you for this day. Thank you that we can participate in this class, and please bless that we'll use what we learned today in our daily lives, and please bless that we'll be safe, and please bless all the missionaries in the mission field that they might be safe. And say these things in the name of Jesus Christ. Amen.
Amen. Thank you very, very much.